So I'm Doorgesh, and I'm a biomedical scientist. And today the project that we are going to discuss is about basically developing new systems using AI and other technologies to enable early diagnosis of different types of tumors. And that is still a big challenge in the field of biomedical science. And that's the problem we are trying to tackle right now.
Okay, and could you tell us more about this problem now? Is that about detecting faster tumors and what type of tumors? And could you give us a bit the challenges around this topic today?
Correct. So one of the main challenges of early detection with respect to cancer is that at the early stages you have very few cells which are cancerous, and they are usually surrounded by a huge pool of healthy cells. And it is very difficult for pathologists to identify this small number of cancerous cells, surrounded by millions, literally millions of healthy looking cells. Of course, pathologists are trained to do that and they do it quite well, but the problem is they are humans and there's a limit to, how many samples they can look at or to what detail they can analyze the samples. So, of course, we can't expect a pathologist to look at every single cell, in a pool of 5 million for one patient. So that is where I think computers can help, because computers can do this very well. And computers don't get tired. They can work, 24 hours. And they, more and more, it's becoming clear that they do a very good job at this single cell analysis. So that is one aspect that we've tried to address. The second aspect is, how do we identify cancer cells? So there are several methods for that. And for example, doctors look at the nuclear size. Is it too big? Is it too small? Is it too dense? And so these are some features that doctors, pathologists who are well trained can identify. And so but usually to do that, you need to process the sample a lot. You need to stain it with different types of dyes and all that. And what happens then is, when you've identified the tumor, you've lost the sample because the sample was degraded. So you can't do downstream analysis with that. So one of our aims was to be able to do this without degrading the sample, so that in case we identify a disease, can we do further tests on the same sample to help with the treatment? So that's another thing that we've tried to address.
And I've read in one of your articles that thanks to you're not only able to detect if there is a cancer, if there are some cancer cells or not, but you can also detect the stage of the cancer with some metastases, or if it is a benign tumor. Could you tell us more about how accurate you can be, in the stage identification, and how was it done before? I mean, how much time did it take before to understand, where the patient were in his disease actually?
Yeah. So traditionally, this work is done by a very experienced, well-trained pathologist, where they will look at the slide of a biopsy slide, and they will analyze each and every portion of it. They will look at, the size of the tumor and they will stain it with different type of markers, protein markers, and then depending on that, they will determine what stage it is at. What grade of cancer it is. So now that is, of course, a very lengthy process, first because of the sample preparation itself, and also because the pathologist has to spend a lot of time to do that. So what we have known for a long time is that the cancer progression it's a progression, it's a progressive disease. So it starts at some point and then it keeps changing. As it evolves, there are changes which are accumulated in the cells. So we've tried to see whether we can use these, these trajectories, these changes that keep happening, this pattern of changes, to pinpoint which stage the cancer is right now. At the moment of the analysis. And our machine learning model seems to be doing quite well. Not as well as identifying a cancer cell from a normal cell, but so the accuracy right now for differentiating between different types of cancer cells is around 70% at single cell level, and based on our pH imaging. So, however, now that we are going forward in this direction, we are going to focus more on trying, to distinguish these different grades. And we already have a few ideas in mind of how we can improve this accuracy. So instead of using just the pH information that we get, we can also incorporate structural changes that happen as the cell progresses.
And this new technique is applicable to what type of cancer? I've read about breast cancer, liver cancer. What do you have in mind, and what are the limits of this technique for some specific type of cancer, actually?
Yeah, so we've when we started with breast cancer, and so that is what the initial project was based on. But then soon we realized that it would actually be very useful for liquid tumors. Okay, because what we can now think of is if we have a channel in which we can flow the cells. So for example, a blood sample from a patient, and we can image each cell as they are flowing in that channel. In the background, we can also analyze each individual cell. So that is something that we don't have right now in the medical field. So then we thought that, okay, maybe we should focus on liquid tumors, and we've started some collaboration with a few clinicians where they think it would be very useful for bladder tumors, because in bladder tumors, what happens is invariably you have a few cells which detach from the bladder and get in the urine, and that can be analyzed in a very non-invasive way. So that is, I think, one direction in which there is a lot of potential. And as far as the limitation is concerned, so right now, of course, the big problem we have is lack of training data. So what we would want to have is, patient sample from different patients from different countries, different races, different, so that we can incorporate all the variability in our models. So right now, of course, that's not there just because it doesn't exist. But there are a lot of global efforts going on to build such data sets, and I'm sure in the near future we will have better models just because we have more data to train on.
And I guess it's the dream of any data scientist.
Of course.
And I guess it's a good transition. Maybe if you could tell us about the technical pipeline that you have implemented, and what are the different steps, generally speaking, to identify the pH of the different cells, and in the end, of course, what are the percentage of cancer cells, etc. Could you tell us about, in a technical point of view, you have implemented the different computer vision steps?
Okay, so the first step is, of course, sample collection. So we get the sample either from a lab or patient, or it doesn't matter, from any source, even animal models we can take. Then what we do is we have to stain the cells so that they can be visible in the microscope. So that's a very quick step. It's around 15 to 20 minutes. And then after that is the imaging part. So with the imaging right now, we are using a bright field microscope, color microscope, so that we can, because the pH is reflected as a color on the microscope. So is the imaging part. Then comes the more computer vision aspect, where we have to segment individual cells, because you can have, in a sample, you can have 5 cells, or you can have 5 million cells. And so we have developed a few computer vision tools to segment individual cells. Then we do the feature extraction. So from each cell, we extract different features. So what we've known, again, for a long time now, is that within a cell, the pH is not uniform. Different organelles, different regions of the cells will have different pH depending on their functions. And in cancers or in other diseases as well, the maintenance of this pH is disturbed. So what we are trying to do now is, if we can get an image, a map of this pH distribution, can we use that to know whether this map corresponds to a normal pH distribution or a disease pH distribution? So we extract all the features, and then comes the machine learning part, where we train basically the model. Okay, these patterns are associated with healthy cells, this pattern are associated with cancer cells. So that's the general pipeline right now. Maybe I can just add, going forward, what we plan to do is, instead of getting everything on a slide, if we can get them in a flow channel, that's another step forward, because then we can also separate the cells, whether they are healthy or not, put them in different channels, and do downstream tests with them.
Okay, no, that's pretty clear. Perfect. And just before you mentioned in this regard that one of the limit to maybe bring this technology at full scale and detect other type of cancers, you might be limited by the quantity and the quality of data, probably. For this project and for further projects, are there any plans to work with other labs, other companies, to increase the size of this data set, and how do you label, how do you deal with this data problem actually, and the privacy maybe as well?
Yeah. So there are a few ways. So first of all, right now, to begin with, what we have to do is we just take known cell lines. So these are commercially available cell lines. We have them in the lab. So we know this is cancerous, this is normal, both are from the breast, and so we can use them. Then this is the first step. Now, once we've concluded that, okay, our technique is working well on this, the next step is to take either primary cells or primary cells, meaning from patients from hospitals, or animal models. So one important aspect is to convince the medical practitioners, okay, because they also eventually is going to be useful for them and the patient. So they have to really believe that it works. So what we started doing is we started to collaborate with clinicians. So where they can provide us sample that they are working on, that they know, and we can then show them that, okay, the results we are getting matches what you are, identifying on your side. So that is also a very small step forward, because we still are not able to get samples from, thousands or millions of patients, from different categories. So that's something that we haven't tackled yet, but we are trying to see with different hospitals whether we can get samples from them, and this is an ongoing process which hopefully will bear fruit soon.
I've read that this technique allows detecting cancer in less than 35 minutes. Could you give me the time as of today, to detect with practitioner, visualizing the slides, etc., how much time it takes, and what are the benefits for the patient? Because do you have an idea to quantify how much people can be saved or can cure faster from cancer? Do you have this kind of metrics, or it's way too early to quantify?
I mean, it's still early for me to tell you how many patients we can save, but in terms of time, I can definitely tell you. Right now, in a conventional way, if you take a biopsy, if everything goes smoothly in the traditional pipeline and the pathologist is free at this time, as soon as your sample comes, they label it, analyze it, and give a report, it'll take a couple of hours easily. Actually, just the sample preparation itself will take a couple of hours. Plus now the pathologist analyzing it. So easily a day at minimum. So now, of course, in the huge system that we function, you'll have, thousands or hundreds of patients, and all these samples have to be prepared. Then the pathologist will look at them. So it will take a few days in the conventional system right now. And what we are offering also, so right now, of course, in the lab condition, it's taking 35 minutes. I imagine in a hospital setting, it might take a few hours, but that's still a huge leap from a few days. And the way it will benefit the patient, there are actually a few ways in which it will benefit. First of all, what we are arguing for is that for the pathologist, it's usually more difficult to identify early stage cancer, and that's well documented. Okay, pathologists will most of them, I won't call it mistake, but most of the misses will happen at earlier stages. So what we are proposing is a system which is going to focus more on these early stages and basically guide the pathologist that, okay, here we think, there's a region which you need to put more focus on and all that. So that's one way the patient will benefit. And the second way is, as I said, our cell is not killed in the process. So if we identify some a disease, we could then use these cells for downstream testing. For example, test different types of drug. Right now, if you're diagnosed with cancer, the doctor will of course take into consideration different aspects of the patient and decide which drug is the best. Okay, but what if now we could investigate those cells which are coming from that specific patient to tell us more about which drug would be more effective? So that's another direction in which we are thinking more in terms of personalized medicine.
That was a great conclusion. And thank you very much for your time, Doorgesh.
Thank you very much for your interest in this and also to bring this to your audience.
Awesome. Thanks a lot for your time.